know what color blindness, hairless cats, and deformed strawberries have in common? You guessed it, gene mutations. What are gene mutations? Changes to the genetic makeup of a cell. Gene mutations are important for the diversity and the creations of new traits in living things. In this segment, we will be going into depth on small-scale mutations, known as point mutations. A point mutation is a change in a single nucleotide pair in a gene. There are different types of point mutations: single nucleotide pair substitutions and nucleotide pair insertions or deletion. Single nucleotide pair substitutions include silent mutations, missense mutations, and nonsense mutations. Nucleotide pair insertions and deletions include insertions and deletions.
The first mutation we will dive into is silent mutations. Silent mutations do not have an observable effect on the organism, hence the name silent. It is unobservable because of the specific type of substitution that occurs. Looking closely, a single nucleotide base in the sequence is substituted for another nucleotide base. Although there is a change in the sequence, the amino acid that is coded for the new sequence is identical to the original sequence or has similar properties to the original, resulting in no noticeable change in the protein. To reiterate, silent mutations have no significant change on the properties of the amino acid and the protein as a whole. The next type of substitution is a missense mutation. Missense mutations can have a significant impact on the properties of the protein that is coded for the amino acid sequence. A missense mutation occurs when one nucleotide base is switched with another nucleotide base. Unlike silent mutations, missense mutations result in a change in the amino acid that is coded. Very often is this change insignificant and does not play a vital role in changing the protein's function. However, if the substitution falls within a crucial region in the protein, such as the active site of an enzyme, the mutation may result in significant consequences that change the protein's activity. An example of a missense mutation is a disease that many have heard of, known as sickle cell anemia. This disease results in the sickling of hemoglobin. This sickling is caused by a mutation in the beta globin. In the mutant beta globin, the CTC changes to CAC on the DNA template strand. The mutant mRNA then has the codon GUG rather than GAG. Because of the substitution, the codon that coded for glutamic acid now codes for valine. The missense mutation causes sickling of the red blood cells, which can cause frequent blood clots and lack of oxygen in the blood.
Nonsense mutation is the last type of single nucleotide pair substitution. A nonsense mutation is the most detrimental type of substitution. This substitution results in a premature stop in the translation of a protein. The polypeptide chain that results is significantly shorter than the polypeptide of the original gene. Most of the time, nonsense mutations cause non-functional proteins. An example of a nonsense mutation is cystic fibrosis. Although cystic fibrosis can be caused by other mutations, nonsense mutations are common causes. Cystic fibrosis results when the cystic fibrosis transmembrane conductance regulator protein, or the CFTR protein, is not made properly or is not made at all. Nonsense mutations within the CFTR protein cause its production to stop prematurely. This mutation results in the inadequately made protein, which causes the buildup of thick mucus in the lungs and affects the digestive system. Those with cystic fibrosis tend to have a shorter lifespan, but with modern-day medicine and medical practices, lifespans have significantly increased.
The next type of mutation is nucleotide pair insertion. This mutation adds one or more nucleotide base pairs to the sequence. If the number of bases is not a multiple of 3, then it is also called a frame shift, because it changes all the codons after the insertion. A disease resulting from insertion is Huntington's disease. Normally, the CAG segment repeats 10 to 35 times, but people with Huntington's disease have it repeating up to 120 times. Finally, we have deletion, which is a mutation that removes one or more nucleotide base pairs. A disease resulting from deletion is 22 deletion syndrome. caused by deletion of some bases in chromosome 22. This disease is characterized by cleft palate, heart defects, and autoimmune disorders. The longer deletions and insertions usually occur when one part of the chromosome crosses over and changes genetic information with another chromosome. Frameshift occurs during insertions and deletions. Unless the frameshift is very close to the end of the gene, the protein is usually non-functioning.
Thank you for watching this week's video on gene mutations. Tune in next week for gene regulation. And as always, stay mutating!